Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we discussed about the principles, the initials of DNA biotechnology or recombinant DNA technology, in which we came to know about restriction enzymes, different other enzymes, what are the different tools which are required in genetic engineering. Okay, we came to know about palindromic sequences, the sticky ends, the blunt ends, different type of restriction endonucleases we discussed, and their action and their cleaving site where they basically attack. Where what is the recognition sequence and the site of cleavage? Okay, so my dear students, I will tell you again that recognition sequence is basically that site which is recognized by the restriction enzyme, the restriction endonuclease, and the different types of restriction endonucleases. Especially type 2 restriction endonuclease is used to make rDNA, recombinant DNA. Okay, so cleaving site or the recognition sequence is that site which is identified and recognized by the restriction enzyme to make a cut. That cut may be a sticky cut or cohesive ends, or it may be a blunt end. Okay, so today we are going to start with a very important topic, that is gel electrophoresis, which is based on the separation and isolation of DNA fragments, RNA, and proteins. So gel electrophoresis, it is used to distinguish or used to separate DNA fragments, RNA fragments, and proteins. After the cutting of DNA in which the gene of interest is there, with the help of restriction enzyme, we use this technique, gel electrophoresis, to separate the desirable gene from non-desirable gene. This technique was first developed by A. Tiselius in 1937. You can see a diagram which is given, in which we are having a gel tank with agarose gel, okay, and a power supply, a DC power supply, which will provide a electric field, Okay, agarose is one of the most commonly used matrix. It is the most commonly used matrix. Okay, it is obtained from seaweed. Obtained from seaweed, and most importantly, agarose is the polysaccharide. Agarose is the polysaccharide. Okay, so now we will look into the technique. Moving to the next slide. Yeah, this is the separation of DNA fragments. You can see the large sample, the medium sample, and the small sample, mean depending upon size and mass of the DNA, RNA, or protein. So separation of DNA fragments is it basically involves a principle that all naturally occurring colloids, they are having a specific charge. Okay, so as you have studied in chemistry and physics, where the negative terminal, that is cathode, will attract the positive particles, positive colloidal particles, and the positive terminal or the anode will attract the negative colloidal particles. Okay. So positively charged molecules, they will move towards cathode, which is negatively charged electrode, and the negatively charged molecules, they will move towards the anode, which is a positive electrode. The moving medium, okay, mean the coating of agarose gel is done, okay. So we will be needing a matrix or a medium through which the fragments will move. So the medium which we have taken, that is agarose gel, is stationary phase, and the DNA fragments, they are moving phase. Or we can say that the stationary phase or the gel is adsorbent, and the DNA fragments, they are adsorbate. Okay. Now, the gel electrophoresis is the laboratory technique used to separate and isolate proteins or DNA fragments based on mass or size. Samples are placed in a block of gel and an electric field is applied which causes samples to move through the gel. So moving of colloidal particles to their oppositively charged plate is known as what? Electrophoresis. Electro plus phoresis. Phoresis means movement. So it is movement towards electric field. Smaller samples, they are less impeded by the gel matrix, mean they or their adsorption is weak, and the larger segments, they will tightly adsorb. So the smaller samples, they less impeded, mean their adsorption is weak by the gel matrix and hence move faster through the gel. This causes samples of different sizes to separate as they travel at different speeds. So the gel electrophoresis is the laboratory technique used to separate and isolate proteins or DNA fragments based on mass and size. Samples are placed in a block of gel and an electric current is applied which causes the samples to move through the gel. Smaller samples are less impeded when adsorption is weak by the gel matrix and hence move faster. Larger samples, their adsorption is strong. This causes samples to to distinguish or to disintegrate according to the sizes, and they have a retention factor. A retention factor means their moving velocity on the stationary phase is different. Okay. While both DNA and proteins are separated according to the same basic process, the basic process is same, but protocols are different. Okay. DNA may be cut into the fragments using restriction endonucleases. Different DNA samples will generate different fragment lengths. Okay. So the DNA fragment length will be different as cutting from different restriction endonucleases. Fragments separate because DNA is negatively charged due to the presence of phosphate group, as DNA is a negatively charged colloidal particle as it is having a free phosphate group on each nucleotide. Okay. So it is a negatively charged colloid. DNA samples are placed into an agarose gel and fragment size calculated by comparing the standards given by the industry. Mean every industry or every company, whosoever is manufacturing the gel electrophoresis machine, they have their own standards and we have to work according to those standards. The specific sequence can be identified by incorporating a complementary radio-labeled hybridization. Once the fragments are separated, Now, to separate a specific gene of interest or to separate a particular fragment, we need that fragment having a single stranded DNA base pair to get a complementary base pair. The moment complementary base pair is attached to the gene of interest on the agarose gel, the bond will be formed, the hydrogen bonding will be formed. So specific sequences can be identified by incorporating a complementary radio-labeled hybridization probe, transferring the separated sequences into a membrane, and then visualizing by autoradiography. This technique is known as was Southern blotting. Okay. First, what we have to do, we have to separate by restriction endonuclease. Then the electrophoresis is done, the segments are separated. Now we will make a DNA probe. That DNA probe is like a pen. Okay. So that DNA probe, when this pen is run over the agarose gel or the membrane, okay, so what will happen, it will stick to the complementary base pair. So the compatible complementary base pair of gene of interest, if it is having the complementary base pair with the probe, then they will attach and they will make a hydrogen bond. After that, they will be transferred to the membrane and then they are visualized under autoradiography, which is known as what, Southern blotting. Separated DNA fragments can only be seen either after radio labeling them, okay, or by treating them with ethidium bromide, followed by the exposure to UV radiations. So the DNA fragments which are separated, after getting separated from the DNA probe, then they are treated with the ethidium bromide followed by exposure to UV radiations. UV radiation give them a orangish color. Okay. So because of UV radiations, they give a bright orange colored bands. Bright orange colored bands. Gene of interest. Now, after finding the gene of interest, we have to take out the gene of interest from the gel. So cutting out the DNA fragments means removing the adsorbent. This process is known as what? Elution. The removal of adsorbent is known as what? Elution. Elution is a term you have already been familiar in chromatography. In chromatography, the same technique is here only. The chromatography is happening only because colloidal particles and the movement of colloidal particles under the electric field on a stationary phase is the chromatography. So cutting down of DNA fragments, okay, from the agarose gel. So first we have to cut down the agarose gel from the plate. So after cutting down the agarose gel from the plate, then we have to snatch out the DNA or the gene of interest with the help of DNA probe we are having. So this process known as what? Elution. Okay. After taking out the gene of interest, it is introduced into the chlorine vector for further processing. Protein separation. Proteins may be folded into a variety of shapes like you have studied in class 11 biomolecules, okay, secondary, tertiary structures, quaternary structure. And that is why because of infolding of protein and the amino acid molecules because of intensive inter and intramolecular hydrogen bonding, which affects the size and have positive and negative regions, no clear charge is there. Okay. So proteins may be folded into a variety of shapes which affecting the size and have positively and negatively regions, no clear charge. Proteins must first be treated with anionic detergent. We take a anionic detergent, sodium dodecyl sulfonate. So this is the anionic detergent sodium dodecyl sulfonate in order to linearize and impart a uniform negative charge. So this sodium dodecyl sulfonate will give whole of the colloidal sample of protein a negative charge, a uniform negative charge. Protein samples are placed into polyacrylamide gel. Then we take a polyacrylamide gel and protein samples are placed into it and sizes compared against the known industry standards, as we were doing in agarose gel electrophoresis for DNA. So polyacrylate gel electrophoresis for protein and agarose gel electrophoresis for DNA. Southern proteins are transferred to a membrane, and then target proteins are identified by staining with a specific monoclonal antibody. Monoclonal antibody, suppose it is like this. This monoclonal antibody, it is having a antigenic determinant site. Suppose this is the antigenic determinant site, and here is the protein. So if this protein, this is the protein, this protein is bonded to the antigenic determinant site, then yeah, it gives to the desensitization or the identification. So monoclonal antibodies, they identify the protein or a product, proteinaceous product, okay? And after staining it with monoclonal antibody, this process known as what? Western blotting. So please make out very sure that DNA, when we talk about ELISA, okay, and if ELISA is done for finding the DNA, confirmation of DNA gene sequence, then Southern blotting is the confirmatory test. And if we are finding out a particular protein or immunoglobulin, whether it is present or not, then we will go for Western blotting. So confirmation of specific protein is done by Western blotting, and confirmation of specific gene sequence is done by Southern blotting. Okay. This is the gel electrophoresis for protein. You can see the gel electrophoresis, different, gel transfer, antibody staining. Okay. Fluorophore mean it is also a radio labeled monoclonal antibody or stained, okay, antibody. Electric charge separates the proteins according to the size. Proteins transferred to the solid support of membrane. Proteins stained with specific antibodies for visualization. So this is the process of gel electrophoresis for proteins. I hope you got it. So my dear students, this is how the gel electrophoresis, polyacrylate gel electrophoresis is done for the separation of proteins and DNA. So my dear students, we will be discussing about the cloning vectors, their types, okay, and especially PBR322 in detail in the next class. Till then, bye-bye, take care, stay safe, stay at home, happy evening.